Molecular cloning. Molecular cloning refers to making numerous copies of DNA fragments of interest, such as a gene. Molecular cloning is a set of techniques to insert recombinant DNA from a prokaryotic or eukaryotic source into a replicating vehicle, such as plasmids or viral vectors. Techniques to be discussed will include plasmid purification, restriction enzyme digest, DNA ligation, and transformation.
Bacterial library. A collection of bacterial cells that has been transformed with plasmid vectors, carrying random DNA inserts from a single species. A small piece of DNA taken from a virus, a plasmid, or the cell of a higher organism that can be stably maintained in another organism, and into which a foreign DNA fragment can be inserted for cloning purposes. Cloning, the duplication and propagation of a cell or organism. Vector, an independently replicating self-DNA molecule. Why are plasmids used as vectors? Plasmids often carry genes that benefit the survival of the organism, such as by providing antibiotic resistance. Artificial plasmids are widely used as vectors in molecular cloning, serving to drive the replication of recombinant DNA sequences within host organisms.
Plasmid purification. During plasmid purification, bacterial cells are lysed, freeing DNA and other cellular components from the cell wall. Cellular components are then removed, and the DNA-containing lysate is processed to further remove contaminants and separate the plasmid DNA from the genomic DNA.
Bacterial colony, a clump of genetically identical bacterial cells growing on an agar plate. Because the cells in a colony are genetically identical, they are clones.
DNA ligation. Ligation refers to the joining of two DNA fragments through the formation of a phosphodiester bond. An enzyme known as ligase catalyzes the ligation reaction. In the cell, ligases repair single and double-strand breaks that occur during DNA replication. Molecular cloning is a method to prepare a recombinant DNA molecule, an extrachromosomal circular DNA that can replicate within a microbial host. DNA ligation is commonly used in molecular cloning projects to physically join a DNA vector to a gene of interest. The ends of the DNA fragments can be blunt or cohesive and must contain monophosphate groups on the 5' ends. Moreover, the covalent bonds are formed and a closed circular molecule is created that is capable of transforming a host bacterial strain. The recombinant plasmid maintained in the host is then available for amplification prior to downstream applications such as DNA sequencing, protein expression, or gene expression. The polymerase chain reaction, otherwise known as PCR, is commonly used to amplify a gene or a DNA fragment of interest from any source of DNA to be cloned. In order to generate compatible ends, it is common to add restriction sites to the 5' end of both PCR primers. When adding restriction sites to a PCR primer, it is recommended to include 6 bases between the recognition site and the 5' end of the primer. These additional bases provide sufficient DNA for the restriction enzyme to bind to the recognition site and cut efficiently. When selecting a restriction site to add to the primers, it is important to determine which site will be compatible with your selected vector, whether directional cloning is desired, and, most importantly, confirm that the recognition site does not occur within the gene or DNA fragment. Transformation. In molecular biology, transformation is the genetic alteration of a cell resulting from the direct uptake and incorporation of exogenous genetic material from its surroundings through the cell membrane. Bacteria can take up foreign DNA in a process called transformation. Transformation is a key step in DNA cloning. It occurs after restriction digest and ligation and transfers newly made plasmids to bacteria. After transformation, bacteria are selected on antibiotic plates. Key steps in the process of bacterial transformation are the following. 1. Component cell preparation. 2. Transformation of cells. 3. Cell recovery. And 4. Cell plating.
Steps of molecular cloning. Step 1. Isolation of DNA fragments to be cloned. Before we carry out the operation of gene cloning, we need 2 basic things in their purified state: the gene of our interest and the vector. Similarly, the vector is a carrier molecule which can carry our GI into a host and replicate there along with the GI making its multiple copies. In this state, the GI can also be expressed in the host cell producing the product of the gene, which is needed by us. Step 2. Insertion of isolated DNA into the suitable vector to form the recombinant DNA. Our next step will be to cut both the vectors as well as the GI by using a special type of enzyme called restriction endonuclease. A restriction endonuclease is an enzyme that cuts double-stranded or single-stranded DNA at specific recognition nucleotide sequences known as restriction sites. After this, we move to pasting. Here, the GI is taken and pasted into the cut vector. This procedure also needs a binding enzyme called DNA ligase. The resulting DNA molecule is a hybrid of two DNA molecules, our GI and the vector. In the terminology of genetics, this intermixing of different DNA strands is called recombination. Step 3. Introduction of the recombinant DNA into a suitable organism known as the host. When our recombinant DNA molecule is ready, we need to introduce it into a living system known as the host. This is done either for one or both of the following reasons. A. To replicate the recombinant DNA molecule in order to get the multiple copies of our GI, or B. To let our GI express and produce the protein which is desired. Introduction of the recombinant DNA into the host cell is done in various ways and strictly depends upon the size of the DNA molecule and the nature of the GI. Some of the methods followed to carry out this step include electroporation, microinjection, liposuction, etc. Step 4. Selection of the transformed host cells and identification of the clone containing the gene of interest. The transformation process generates a mixed population of transformed and regular host cells. As we are interested only in transformed host cells, it becomes necessary to filter them out. Step 5. Multiplication/expression of the introduced gene in the host. Once we have purified our transformed host cells by the screening process, it is now our job to provide them optimum parameters to grow and multiply. In this step, the transformed host cells are introduced into fresh culture media, which provide them rich nourishment, followed by an incubation period. Step 6. Isolation of the multiplied gene copies/protein expressed by the introduced gene. In this step, we isolate our multiplied GI, which is present, with the vector or the protein encoded by it. Step 7. Purification of the isolated gene copy/protein. After the harvesting of the isolated gene copy or the protein, it is now our job to purify them.
Relevant research from the Journal of Biological Research. Over the last decade, cloning has transformed biological science. In this research, the polymerase chain reaction was put into plasmids and cloned. This was to test to see if using different restriction enzymes and primers would produce more of the TD tomato fluorescent gene. This is just one example of the use of molecular cloning. Thanks for watching!